There you go.
Hi, I'm Lorraine Twardock. I'm an independent toxicology, non-clinical toxicology consultant, so I think I'm an odd bird in the group. I have a question for Dr. Munshi about the bioequivalence studies. I notice, and this might apply to several speakers as well as speakers from yesterday if they're still here today, there's an increasing trend towards the use of biologics as actual prescription drugs, which would eventually wind up as generic drugs. And in many of these substances, there's no identified API, so that a lot of the methods that everyone's been talking about do not apply. Has any thought been given to the types of methods to assess bioequivalence for these types of compounds?
So as far as that goes, I think you're talking about large molecule proteins, things of that nature. Office of Bioequivalence, we work on small molecule products, so I don't think I could really answer that question.
Yeah, now they're getting on the market as supplements, so avoiding FDA scrutiny.
Thank you. Microphone number 2, nice and close for me, please.
Thank you. Hi, I'm Kandasamy Subbaraj from Lupin Pharmaceuticals.
A little closer to the mic, please.
Sure. Kandasamy Subbaraj from Lupin Pharmaceuticals. My question is regarding the PFC requirement. The PFC requirement guidance says it has to be 3 approved products, meaning that, that's what the question here, the 3 approved products including generic plus 2 generics, 1 RLD, or generics plus 3 RLD plus 3 generics. That's the question. Because we have an instance where there was a confusion, so.
A moment while the panel ponders. Okay, somebody has to lean forward in their chair, so.
So is your question specific to the priority aspect of no more than 3 approved?
No, the selection of PFCs, when we submit application, so you grant the PFC status for that application, there was a confusion. It says, the PFC guidance says 3 approved products. That meaning that, are you saying that already like 1 RLD and 2 generics, or you are saying 1 RLD, 3 generics?
Is the question that the PFC guidance says 3
approved
but
products.
The map for expedited review prioritization says something slightly different? Is that the question?
It says 3 approved products, 3 or less, so you'll get a PFC status.
Right, that's the priority determination.
1 RLD plus 2 generics, or 1 RLD plus 3 generics.
I think there might be some confusion because I think it's I can't speak for the PFC guidance, but if we're talking about for a PFC to be eligible, it has to be a priority. And according to the map, the priority map, it's no more than 3 approved products.
Right, that 3 approved is 2 generic plus 1 RLD, or 3 generics plus 1 RLD. That's the question.
Yeah, let us defer and get back to you on that one.
Sure.
Okay.
Thank you.
That was a toughie. For the sake of time management, we'll move on. Again, and let me remind all the folks on line especially, we probably won't have time to get to all your questions. If you're left with any questions, you see the email address on the screen where you can send your questions afterward to be answered. Let's go with an online question if we could, please.
This is directed to Commander Sansone. At what point in the review of a PAS will the applicant be notified that an inspection has been triggered and the later GDUFA goal date, either 8 months or 10 months, will now be used?
So when a PAS is you know, if you submit a PAS, it's acknowledged with actually 2 goal dates. You actually get the 6 month, just for example, you get the 6 month for a goal date if an inspection is not required. You get the 10-month goal date if an inspection is required. So you actually get both goal dates. We can't alert as to inspection is required until we actually know it's coming and has been deemed required for inspections. So you'll actually get 2 goal dates.
Okay, microphone number 1. Nice close for me, please.
Yeah, my name is Mizia from Allen Laboratory. I asked that question yesterday, but since this question is for Dr. B.J. Patel for filing review, I repeated the question yesterday. Since you covering your topic this morning, I want get a clarification or also the understanding for the extended reach product. If the product specific guidance does not mention alcohol study, is that required for the applicant submit alcohol study for that particular product? Also, in your same topic, mention the half-score tablets needed required for comparative dissolution study in multi-medium. If the product is not intended for administration for half tablets, there's no score on the tablets. Is that required for the half tablets dissolution on the half tablets in the multi-medium dissolution, multi-medium dissolution study?
Oh, yeah, I'm sorry. Yeah, hold down that first button for a second. There, you got it.
So you asked multiple questions. So your first question was, is alcohol dose dumping required
if
at
it's
filing if
yeah.
it's not identified in the product specific?
Yes, correct.
Okay. If the product specific guidance doesn't identify alcohol dose dumping, that's not something that at the time of filing you would look for. When it gets to the technical review, Office of Bioequivalence may ask for it, but at the time of filing, if the product-specific guidance doesn't explicitly state that alcohol dose dumping is necessary, then it won't be a filing consideration.
Got it. Thanks.
With half tablet, if multimedia is requested in the product-specific guidance for that drug product, then half tablet dissolution should be conducted in the multimedia.
If not required, if it didn't specify in the product,
then no. So the dissolution recommendations are based upon what's in the product specific guidance and what's in the dissolution site.
Got it.
Okay.
Thanks.
Yep.
All right, microphone number 2. Nice and close for me, please.
Hi, this question is related to filing issues. My question is, you said there are some RTRs given based on the impurities. So you said that is lack of justification for the impurities. So do you consider inadequate justification of impurities also as a reason for RTR? And other thing is related to failing accelerated stability data. If the applicant is not basing their initial submission based on 6 months data and they are submitting only 12 months at the time of submission, does the agency expect 6 months accelerated, failing 6 months should also be submitted? I know they expect it, but why that is needed if applicant is basing its initial submission based on 12 months of data?
Can you repeat your first question?
First question is RTRs given based on lack of impurity justification. Say justification is there, but it is inadequate. Do you consider that one also a reason for RTR?
So inadequate justification is specifically for unspecified impurities. The RTR guidance states that for unspecified impurities, a justification of USP is unacceptable. So if you provide a justification for any other impurities, if you provide a justification, then from a filing perspective, we would take it. However, for unspecified impurities, and it also states it on the model tables that OPQ provided for impurities, it states in the table that if for an unspecified impurity, if the regulatory AC is greater than, IC is greater than the QT.
IT.
AC is greater than regulatory IT. I'm sorry. Then justification isn't, like, you can provide a justification, but the RTR guidance states that USP is one that's not acceptable.
So like that's only for unspecified. What about the identified impurities? If those are
You
ever
can provide a justification, and that
so-
will be acceptable from a filing perspective.
Oh, okay.
And then your second question was
Like
stability.
it was more stability.
So the recommendations in the stability Q&A state that an applicant should be submitting 6 months of accelerated and 6 months of long-term. So if an applicant submits 4 months of accelerated stability data, then it fails to meet the recommendations for the 6 months of accelerated stability data. And that is why you get a refusal for receipt.
So I'm not saying that one. I'm saying like we have seen the failure in 6 months or 4th month. I don't want to submit until I gain 12 months of long-term data or intermediate data, and then only I'm going to file my application.
Mm-hmm.
So does agency still expect that applicants should provide the accelerated data, or if we are filing the application with 12 months of data, that should be acceptable?
So,
Is that a RTR criteria?
So the expectation is that applicants submit 6 months of accelerated stability data. So even if the accelerated stability data is failing, you should be providing 6 months of the failed data. So if it fails at 3 months, you should continue the stability data to 6 months.
Okay, thank you.
Okay, and just a reminder to the folks in the room, I can't threaten you with having to buy drinks tonight. But please, for courtesy to the other people standing in line, 1 question per microphone, and then you get to go to the back of the line. Online question for me, please.
Sure, this is for Derek. When you said that notify Office of Regulatory Affairs of changes in manufacturing facilities ASAP, what types of changes need to be told to the ORA?
So the focus of the presentation, in line with JD's presentation, was the master facility data. So we're talking about changes in the address, changes in locations. If you have multiple sites on a campus, if you're into a single entity, just make sure that you're updating ORA as fast as possible so that their database, their official database, is updated with whatever information that's going on. And we're talking about the name, the address, the location, the DUNS number, if there's a new DUNS number or something like that, and then that way they can update their database for the FDA establishment identifier, so that way when the application comes in, JJ's group then has consistent information between ORA and CDER that we can then conduct the assessment.
Okay, microphone number 1.
My question is to Saman, and others please chime in. Being in the regulatory area for many years, I've seen inconsistency reviews from the agencies for many, many years, and I'm really thankful to see this IQA, and quality assessment is very good to see that. It's a good stepping. What I have seen is that a company, we can have a same molecule with different dosage forms. What we also are exposed to as sometimes, reviewer asks questions, and we do anticipate relevant question to other dosage forms. And sometimes we don't get it, which is good, I'm not complaining, but sometimes we do get it. And so, with that said, now I'm asking the question, if you guys can elaborate on what is these assessors are doing, how they are assessing, are they looking at other dosage forms, other applicants, how that is being assessed?
So this is something that we are working on constantly, I would say. With the role of the ATL, it will hopefully help in the future to kind of make less discrepancies in these deficiencies. But regarding dosage form, usually we try to keep the team similar. as much as possible depending on the workload. So that is our goal, to do that. And sometimes the workload does not allow that. But hopefully at the end, when the ATL looks at it, he's able to actually narrow down these discrepancies. So hopefully that answers.
Microphone number 2, please.
Okay, my question is for Mavis and Bio. On the Q1, Q2 sameness, the RLD lists 3 excipients, and it says less than X number of milligrams. How do we determine the 95 to 105% based on a less than quantity?
Your question correctly. When you say less than, can you elaborate on that?
It says less than so many milligrams per milliliter for like an excipient, like a thiosulfate.
Okay, so for the Q1, Q2 assessment, for Q2, you have a range of 5% plus or minus. So that's how we would assess the 95 to 105% concentration of the RLD. Does that answer?
So is the less than, like say if it says it's 20 milligrams, but it's less than 20 milligrams, would the 20 milligrams be the 105%? Or would that be considered the 100%? You can go plus or minus?
So you allow the 5% allowance minus or plus. So minus of plus 5% over the 20 milligram. So if the RLD is 20 milligram, you're only allowed to go 5% or under, above or below.
Thank you.
Online question, please.
For those of us new to ANDAs, could you please expand on what Q1, Q2 means?
Topic here, so I'll elaborate again. So Q1 is the qualitative property of the proposed generic product to that of the RLD. So the active ingredient and the inactive ingredients should match that of the RLD for the Q1 assessment. For the Q2 assessment, the quantities of the active and inactive ingredient should match that of the RLD with the allowance of the plus or minus percent, or 95 to 105% of the RLD concentration.
Okay, microphone number 1.
Keith Weber with Bearr Company, and this question is for Dr. Munshi. Now that there's a guidance on BCS class 3 waivers, question is that those, it seems at this point now that the permeability is not really relevant to the waiver. If you have a product which is highly soluble and rapidly dissolving, that you would get a waiver whether or not it's quickly permeable or low permeability. Is that a correct assumption that we would not need to do permeability assessments?
For BCS class 3, for the guidance, there's no need to do a permeability assessment, but we recommend for BCS class 1 that high permeability be demonstrated.
I guess, I don't know if there's a second question or not, but can you explain why that would be if they're...
Right, so I think the idea being that if it's if it's high permeability, we want to make sure that both the test and that the test product has API with high permeability characteristics, and that risk is a little bit higher for BCS class 3.
I have 2.
All right, let me repeat that. Yeah, so basically, yeah, bottom line is that for BCS class 3, in that case, we consider it lower risk and we don't ask for permeability studies. For BCS class 1, it's higher risk and therefore we'll ask for permeability for a BCS class 1 product.
Thank you.
Microphone number 2.
Hi, this question is for Dr. Munshi. If a major amendment has been filed for a site transfer, manufacturing site transfer, and the bio guidance asks for fast and fed study, which you have done for the original filing. For the site transfer batch, do you have to do both studies?
Generally, it would just be the fasting study. Say if the PSG recommends fasting and fed, generally it would just be a fasting study.
Thank you.
Online question, please.
This question is for Elizabeth. What is the requirement of a contract lab in which they are doing a micro-identification of counts? If there is an objectable organism seen in the contract lab sample, is the contract lab responsible for performing a micro-risk assessment for the sample?
I think I would have to understand better what tests they were performing. You know, if this was something like so I think, you know, it depends on the test they're doing. If they're doing a sterility test, that's obviously a problem. If it's a microbial enumeration of a non-sterile product, let's say, you know, I think then that information may need to be assessed, but actually that might be something that a facility inspector would look at. I think we, on our assessment of the application, I'm not sure that we're looking at that pieces of information. So my assessment may not cover that kind of question. So, Eric, can you comment on if it's a facility issue?
Yeah, so I mean, I think if I'll restate a different way, and hopefully it still makes sense. So really the question is, it's a contract testing lab that receives a sample from a facility, they test the sample, and it fails. The question is, what's the lab's responsibility? You know, obviously the lab should determine, was it a part of their error that caused the failure, you know, first, and then obviously the next step would be then to do the investigation or to alert the facility that sends you the sample, so that they can then do their own assessment, you know, of the failure and conduct an investigation into the deviation. That'd be my best recommendation.
Microphone number 1.
Yeah, hi. My question is to Dr. Utpal Munshi. This is regarding classification of certain drug substances and drug product as BCS class 1 and class 3. Is there a mechanism with FDA wherein the agency accepts the standalone BCS classification of drug substances and drug product rather than including that information in the ANDA application?
So I think your question is there a way of qualifying a substance as BCS class 1 prior to the submission itself?
Right.
Okay. To my knowledge, there's no way at the present time to do that. If you wanted to demonstrate that something was BCS class 1, you'd have to do that through the application itself.
And the possible outcome, if in the opinion of the agency, the substance and the product is not classified as either BCS 1 and 3, the possible remediation from the agency would be to conduct a bioequivalence study for that.
Right. If a waiver is not acceptable, then you would default to a BE study.
Microphone number 2.
My question is again to Dr. Munshi. This is related to the extended release dosage forms where it's only one strength, and you have proven you have a passing bio studies, but somehow you used it. You have a different formulation, and you are not able to match the dissolution with the RLD, but you have a passing bio. Is that a requirement that you have to match the dissolution with the RLD, even though you are not asking for the bio waiver for lower strengths?
Okay, so you're in your paradigm, there's only one strength. Correct. And you are comparing and basically you have a bio study in that one strength. There are no other lower strengths in your example?
That's correct.
Okay. And then your question is, okay, I have a passing BE study, but there's something off about the dissolution data.
Yeah.
Right.
So you have to prove the dissolution in like the range of pH, right?
Right.
So in that one, you are passing everywhere, but you are not able to have a similarity, F2 similarity to the RLD dissolution profile. That
Right.
might be like a different formulation.
Okay, but you're comparing just one test product formulation to one reference product formulation, right?
Yep.
So I mean, in general, in vivo data trump in vitro data. But that being said, first we ask for dissolution data, multimedia dissolution data for the test and the reference product. For your example, we would ask for that, and we look at the data in totality. One can never say that we would totally disregard the dissolution data, but emphasis is placed on the in vivo data in general.
Thank you.
An online question, please.
This question is for Dr. Munshi.
Very popular.
What should a sponsor do when a product-specific guidance changes and your ANDA is pending with the previous requirements that fulfilled it?
So I guess I hearken back to my talk, Regulation 3/2024A, the most reproducible, sensitive, accurate approach. And our guidances reflect the idea or the concept behind that regulation. So in general, I would say that if the PSG changes and your ANDA is in-house, then we would consider the recommendations in the PSG to be the most current and the ones we'd recommend that should be followed.
Microphone number 1, please step up nice and close for me.
My question is related to the refuse to receive related to the microbiology in use stability. So is there a specific number of batches required for this in use stability and for microbiology and for chemistry too?
Do you repeat that question?
How many batches required for the in use stability for micro and
so when you say in-use stability, do you mean an in-use
reconstitution? Yes.
Oh, I see. I see what you're saying. So for the study I was discussing during my talk, where you have a post-constitution or solution study. So for that, I don't know that we have a specific requirement for the number of batches you have to test. I think we would make determination based on what you provided us. There's no strict requirement from my perspective on the number of batches that would have to be tested in that study. It should certainly be a batch that's representative of your manufacturing process for commercial production.
And it's one batch only?
I think it's better to see more, but I think it would be case-by-case determination, but there's no strict requirement for the number of batches in that type of study.
Derek, did you have anything you wanted to add? Okay, just checking. Microphone number 2, please.
Hi, my question is for Dr. Smith. If in the original filing, the DMF holder had listed 2 manufacturing facilities, and during the review, they remove one from the DMF, do we have to update the filing at any point? The other address is still the same, and they may have changed the contact information.
So the DMF has 2 sources. I guess the question would be, in your application, were you using both sources?
Now they're only using one source,
Right.
and that is also listed in the original application.
Right. So if your application lists both sources that were in the DMF removes one of those sources, then your application should be updated to reflect that as well, because you would not be using the source that's no longer in the DMF.
Okay, thank you.
Online question, please.
This is for Mr. Sansone. If the major amendment is for manufacturing site change, Is the PAI of the manufacturing site mandatory?
So let me just to clarify. So for a major amendment, if you're asking if you submit the major amendment, is the pre-approval inspection necessary?
Yes.
Oh, so so yes. So basically, we're going to treat those major amendments the same as any other amendments. So when they come in, we're going to make sure we're going to assess them, see if they need an inspection, and then if an inspection is required, then we'll move from there. So it's treated just like the other amendments that were discussed earlier.
Excellent. Microphone number 1, please.
Here's Keith again. Question for Vincent Sansone regarding the IQA. When multiple applications come in, say P4, you get say 3 or 4 applications come in on the same day and have the same PDUFA lifespan. Do those get assigned to the same IQA team for review or different teams?
I'm going to defer that one to Sal.
So we do have a team identified for a block of, like you can say, multiple entities come together. We will identify them, and we will have a team built for that block.
Thank you very much.
Microphone number 2.
Yep. So my question is related to facility inspection as part of the IQ assessment when the team is formed. So there are 2 goal dates. So at that time, during that discussion, when the team is made, whether the inspection is going to happen for the facilities which are involved in manufacturing or testing, that will be indicated at that time, or that will be informed to the applicant that, okay, now based on the assessment or based on the evaluation agency has done, there will not be any inspection of the sites for this ANDA. And other question is, like, once the DRLs are issued and there are no deficiencies identified, that is coming at the end of 8th month, right, as per the flow chart which you have shown. So after 8th month, if there is a routine GMP inspection, can that have any impact on the final approval of the product or not?
If somebody can remember the first question, let's just do that one for now, and then we'll get to the second question a little bit later. Does anybody remember what the first question was? Can you repeat the first question, but try to do it in 17 words or less?
Oh, okay. So as part of the IQ assessment, the facility which will be inspected by FDA, does that determination made at that time? If it is made at that time, then whether that is notified to the applicant after 2 months of the submission or later.
Answer this, but he can correct me if I misstate what he said. If an inspection is determined, we would not notify the applicant that there will be an inspection. I think Vince said that there will be 2 goal dates that are provided to you in the filing letter, and if there's an inspection, it would be the latter. If there's no inspection, it would be the shorter.
We have to receive the amendment, and then we have to make the assessment to see if the inspection is going to be needed or not. So that takes a little bit of time. So that's why you get the 2 dates. We don't know which one it is until we actually get into the review to see where the assessment will lie.
Online question, please.
This is for Derek. Since the Drug Master File Guidance and associated LOA template does not include listing manufacturing or sterilization sites in the LOA, and is it possible to source multiple different items from multiple different sites? Wouldn't it be better for the ANDA submitter to reference the sites they use and not the LOA provider?
So the LOA was just a recommendation to kind of keep things consistent, because the LOA will be in the DMF file as well as the ANDA. But I think ultimately, as a sponsor of the application, we would want you to list the facilities that are used in your supply chain. So any facilities that are being used for that drug substance that are in the DMF, ideally will be listed in your application, consistent with the current guidances that are available.
Microphone number 1.
Me again. This question is for Dr. Julie Lee and also biopharmaceutical reviewer. In case of extended risk product and the product specific guidance required for ACOS study again, if a multi-strength involved and the formulation is a common blend, so whether it's, I would like to hear agency's view on whether we can do ACOS study on the lowest strength, high strength, use a bracketing strategy rather than do all alcohol on all strengths. Even this study is very simple, but it requires a lot of.
So the product-specific guidance indicates that in order to waive strengths, if you're doing a bioequivalent study, and typically bioequivalent, the product-specific guidance usually identifies the highest strength, and then you can waive the lower strengths. So in order to waive the lower strengths, you're supposed to provide comparative, so test versus RLD, dissolution in all media that is identified not only on the dissolution site, but also on the product-specific guidance. So bracketing, if an applicant provided bracketing, DFR would refuse to receive the ANDA for failing to provide comparative dissolution in all recommended media between the test and the reference listed drug.
Yeah, my question is if the dissolution in multimedia are provided, but the ACOS study using the lower high strength, whether it's acceptable.
Okay, so even in that situation, if the product-specific guidance indicates that alcohol dose dumping should be conducted, then it's on all the strengths.
Okay, got it. Thanks.
Microphone number 2.
This question is sort of related to the last online question regarding facilities listed in the DMF. If the DMF holder, the API supplier, is using test facilities or their sources of their raw materials, a lot of times that's in the closed part of the DMF, and we don't have access to that. Would it, under those circumstances, be okay to reference the LOA in the DMF, or do we have to try to convince them to give us that information?
So, and I think this was another online question as well that was in the list that I saw. The recommendation is to try to include the facilities in your application as best possible, recognizing that there is some confidentiality questions that may come up between yourself and the DMF holder. From our perspective, we would hope that you would have a quality agreement in place that you would understand, the entire supply chain of your drug, right? And that includes the intermediate facilities and the release testing facilities, and especially the release testing facilities, because yes, for the application, you're doing your own testing of the drug substance, but likely under 211, you're going to probably switch to supplier qualification and use the DMF C of A to release the API material with just an identification test. And so at that point, those testing facilities really are the quality control on the API, right? And you no longer have that control because you're not doing the full testing. So I think it's best for us to have that information available in the application because it expedites the review. So it's trying to work with the DMF holders to ensure that at least the critical intermediates and the release testing facilities, to provide that information to us for evaluation.
Online question, please.
Okay. When an ANDA application, when an ANDA applicant submits data showing that the test formulation is Q1, Q2 to the RLD, but the FDA does not agree, what can be done by FDA and by the ANDA applicant to resolve this issue? What advice do you have?
So in that case scenario, a controlled correspondence can be submitted to the FDA, and we can resolve it that way.
All right. Microphone number 1, nice and close for me, please.
My question is to Smith. My NDA goal date, approval goal date is on April 15th. And if any facility, like manufacturing facility audit has been triggered during the March 15th, by then my application is under endorsement with all closing of all division reviews is adequate. What would be your decision period for the facility assessment? Do you consider the March status or the April status, just recent inspection or the previous inspection?
So, yeah, in conducting the facility assessment within the integrated quality assessment team, we do strive to meet the timelines that were presented. And so, yes, during the endorsement phase, we might have completed the review, but if information changes with respect to the facilities, then we would have to reassess that facility. And so, if between the 30 days of us wrapping up the review and handing it off to the actual action date, if the facility becomes non-compliant, then we would have to reassess and determine the impact on the application decision.
But do you have any provision to the public like us to make the strategy on the like a facility status, what you are deciding, and is there any like what are the on the website what we are finding is very nominal VAA, some minimal information on the facility.
I would recommend that you strive to stay in close contact with all of your manufacturing facilities, whether they're within your own company or a contract site, to make sure that they are adhering to CGMPs, ready for inspection, and that the outcome won't be negative in a way that would impact the approval of an application.
See, for those people, they may feel that they are okay, but may not be okay with the FDA. We cannot rely on the vendors and whatever the information they provided. I am requesting, why don't you create a provision to understand to the industry about on each and facility before we listing on the 350CH. This is a crucial decision for anybody. As you know, you have the goals to get approval. Same way we have our own goals and strategies for a particular products to launch. I'll give some date to launch, but it is may not be possible forever if you end up with this scenario. Assessing at the facility at the endorsement phase, whatever it may change. There are number of facilities listing in the 350cc is such a bunch now. 350cc form attachments with all these facilities.
Yes, I think as the industry has grown, the supply chain has grown more complex, and it does make it more difficult to manage and ensure. But what I can say is
this is the area where we cannot control.
Yeah,
and this is going to be a great conversation for the lunch break, which happens in just 2 minutes. So let me go ahead and get to some of the other questions. Microphone number 2, please.
Hi, this question is for Dr. Smith. Again, apologies for bombarding questions at you. This question is related to 356H form. There were a couple of slides presented during the presentation where you have to list all the API manufacturing facilities and testing lab informations. My question is related to the payers and the annual reports. Since the approval of the application, there are, despite of no changes whatsoever in the approved application, do we still need to list those information on 356H for payers and annual report?
So I'm unable to comment on the payers. As for annual reports, I don't know of specific guidance that requires you to submit the 356H form with the annual reports. However, it might be useful to the agency to understand any changes within your supply chain that may not impact the application. So if you were withdrawing a site or not using a site anymore, that wouldn't require a submission update. It would be helpful to the agency if you submitted some kind of information that provided an understanding of what you're currently using, so that we have a knowledge of the current supply chain. But I'm not aware of any current guidance that requires the 356H to be submitted with the annual reports.
Thank you.
Microphone number 1, and then we'll take one quick online question. Microphone number 1.
Nowadays, we do get 2 good for gold dates communications, and we see one with inspection, one without inspection. And sometimes we get surprised because we just had an inspection 6 months ago. Why? So I see it's a general language FDA may be using it. But the biggest challenge as an industry we are facing is that to be ready for launches. So the notification of the inspection is a very critical point. I'm just in line with everybody here. Because we have to launch, we need 6 months in a preparation, sometimes coming overseas and everything. So the notification of inspection is a very, very critical time point for us to make a determination whether we want to launch this product or not. So this is something, and my understanding is based on the GDUFA 2 commitments, there was an absolute discussion that sponsors would be notified about inspection is going to happen in the middle of the review. So if I'm wrong, but tell me, because that's what I heard. So that is, I am requesting the agency to consider, because 10 months goal time or 13 months goal time with an inspection is still a very short period of time for an organization to be prepared and launch a product with everything meeting all the requirements, because we still have to go through the review. We can get comments from the reviewer, change the specification, do this, we have to do validation, all these things. You know, this is one point agency is not really looking at to providing us a good response to give a generic low-cost product to the patients.
Thank you for your input. And we've got that input. We'll take that to heart. Let me finish up with one online question, please.
This is towards Vincent. Can I file a PFC expedited review after tentative approval for adding a new API site if I have enough time for the launch to ensure its approval within the next 8 months?
So I assume this is a post-tentative approval question, and if you're submitting, you have the right after your post-tentative approval to submit a PFC in lieu of coming up your upcoming submission. And now with that upcoming submission will be just treated just like the amendments goal calculation I alluded to earlier. If your PFC is complete and accurate at the time of submission, then yes, it will receive a shorter goal date.
All right, and with that, I've kept the panel up here a little bit longer than intended. Please help me thank the panel for coming up here and dealing with all the questions.